Okay, we are live.
Hello, world.
So it is a great pleasure for me to introduce Dr. Filipa Godoy today. And we are a bit late, but that is mostly because we had such a great conversation and we're chatting. And Filipa convinced me to start a group in Puerto Rico whenever possible. So just a quick introduction. Filipa is originally from Portugal and she did her bachelor's there. Then she did Erasmus in Las Palmas in Spain, which is in Gran Canaria, because she, as we can all understand, wanted to be close to the ocean. Then she went to Puerto Rico for a PhD, where she studied the microbiome of Venezuelan bird, the Watsin. And as a PhD student during one of the ISME conferences, she finally met with Phil Hugenholtz because she had been thinking for a while to apply for an NSF grant to be a postdoc at JGI at Walnut Creek. And that worked out. So she moved to California and worked first with Phil Hugenholtz and then with Susanna Trench. And after that, she became a group leader in Puerto Rico, where she even started her own graduate program. But then, unfortunately, she lost all of her collections during the hurricane, and it was a disaster. But she was offered by the School of Medicine to transfer, where she is now as the chair of microbiology. And now I let you talk.
All right. Thank you so much for your kind introduction. Hello, everyone. Good morning, good afternoon, or good evening, wherever you are. Um, it's a pleasure for me to be able to give this talk today. The title of my talk is Studying Microbiome Dynamics for Cervical Cancer Prevention in Hispanics. It's mostly focused on one of my studies, although I will briefly give an overview of my projects here in the island. I have no conflicts of interest to declare. I just wanted to touch base on the local COVID-19 numbers for Puerto Rico. We have had 2,100 deaths. We are, as data of today, we have 144 hospitalizations and 197,000 positive cases. According to the Department of Health, we've had about 1 million doses of COVID-19 vaccine being administered in the last 3 months. So as Leticia said, I'm in Puerto Rico for now about 17 years. And before my talk, I just wanted to convey a bit of the Puerto Rico's scientific landscape. We have amazing state-of-the-art equipment and facilities. We have the UPR, the University of Puerto Rico, which is the main public university, has 11 campuses. We are here at the university, the campus of the School of Medicine. And the other big campus is Rio Piedras, which is also in San Juan. Have the molecular building where all the sequencing facilities and instrumentation like nanotechnology, materials. We have also the new cancer center. We have a U54 grant from NCI with MD Anderson, also to study malignancies in Hispanics and in Americans. Another interesting thing for those who would like to work on environmental microbiology, we have the only tropical rainforest in the US. It's called the El Yunque, where long-term ecological research takes place. And we also have an island called Cayo Santiago with the biggest free-roaming rhesus macaque colonies, where lots of universities around the world come and study biology, evolution, neurobiology, microbiology. So we have all the conditions to do great science. And I hope you're excited. And if you want to more information about it, you can always contact me or write to the Biomedical Sciences graduate program directly. The email is here below. So I'm at the Department of Microbiology, and we have from immunology to microbiome, have a variety of programs and research labs. So I've been interested in studying biodiversity for a while, for nearly 12 years. as I studied in my PhD degree looking at the Hawatsin, but mostly because microbes have been around for about 4 billion years, and everything that has developed later on, more complex life forms have been in contact with these microbial communities. So everything that exists today, um, all living things and all surfaces, all man-made structures are colonized by these microbiomes. So it's very fascinating to be a microbial ecologist today. Everything that we look at has dominated is dominated by a microbiome. So I've studied, as Leticia said, I studied the Hawatsin for several years. It's the unique folivorous bird in South America that, and it doesn't really fly well. It spends most of the time ruminating with a huge crop. That's why they call it the flying cow or stink bird. And we've been looking at microbial community composition and capabilities for degrading cellulose, for example. So microbes are everywhere. They're essential to health. And as a microbiologist, I always ask myself three questions. Who is there? What are they doing? And how much of it are they doing? And today we're going to mostly discuss about this women's health project. Who is there and what are these bugs doing? So my lab is interested in looking at microbiome-driven malignancies. It's mainly focused on microbial community dynamics, structure and function in vertebrate animals and humans. We use the microbiology in the interface of ecology and other omic techniques, as most of microbiome labs do. So we integrate DNA sequence data with microbiology, physiology, metagenomics, bioinformatics to answer broad questions on host microbe interactions, but mostly on women's health. We want to understand how these communities are established, how they become resilient, what makes them resilient, and how lifestyle contributes to this resilience or not, and persistence, and how can we limit these ecological perturbations to restore these ecological niches. So some of these projects, I have 6 graduate students. Natalie is working on gut microbiome modulation for effective viroimmune therapy against solid tumors. This is a project with the MD Anderson. Kimil is working on an R21, looking at oral fungal and bacteria in high-risk Hispanics with human papillomavirus infections, and also the role of periodontitis. Eduardo is working on combining omic methods and cytokine profilings with the cervical microbiome to prevent cervical cancer. Luis is working on source tracking oral and anogenital microbiomes in promiscuous rhesus macaques in Cayo Santiago, the island that I mentioned before. Elif, she's from Belgium, and she's working with me and with Jim Ackerman at Rio Piedras, looking at the microbiome of nectar and how changes in temperature of the environment can lead to changes in the nectar microbiome, and thus in the interactions between plant pollinator. And we also have a new COVID cohort that Frances is leading. We want to look at how changes in the gut and in the oral microbiome in patients with COVID symptoms and those that have not had any symptoms. And we also have a new clinical trial. It's the Campo, which we're involved with UCSF and Mexico. Other non-clinical microbiome projects include probiotic drinks in the Caribbean, the leatherback sea turtle microbiome, the microbiomes of crabs from different types of mangroves, black and red mangroves. And we continue a collaboration with Venezuela looking at the microbiome of the screamer bird. And why are we interested in looking at human microbiomes? Because, as you know, we're human ecosystems. We are about 43% human, although we have 100 more times more microbial genes than human genes. So the widely cited 10-to-1 ratio for the amount of cells each one of us has been reduced to about the same, fairly the same, 39 trillion microbial cells versus 30 trillion human cells. And what is interesting is that this unit of evolution is not only the host, it's not only the Homo sapiens, but also the microbiome that is associated with the host, the hologenome. So the main lesson here, and this is Day's 2016 paper, shows that the coevolution, selection, and drift occurs at multiple levels on host genomes, on the symbiotic microbiota, and also on the hologenome as a whole. And that's very important because we know now that humans, as holobionts, have transmitted these microbes through generations over time. So we have bacteria, archaea, fungi living in our human bodies. We carry these microbes and we transmit them through the matrilineal line, transferring this primordial birth microbiota through moms, through babies. And we have done this during several generations. So this vertical transmission has led to the conservation of a phylogenetic signal in humans. So we have the human microbiome transferred through, from moms to babies, and with a common phylogenetic signal among whole humans. So where do our microbes come from? So babies, when they're born vaginally, they acquire their first contact with the microbiome is through birth. So in the vaginal canal, which in a healthy woman is dominated by Lactobacillus. And when a baby is born by a C-section, the bacteria that it acquires, it's the bacteria in the skin or in the gloves of the doctors. And as you see here in a paper from Dominguez Bello, also a mentor of mine, she showed that the microbiome from babies delivered by a vagina, they had similarities with the vagina of their moms, which is darker red circles. And in the blue group here are the babies that were born by a C-section, which get the microbiome very similar from the skin of their moms. So definitely this brings questions into the development of this primordial microbiome and the impacts that it has on the immune system and on the baby's development. So basically the development of the human microbiome occurs at the delivery time, whether it's vaginal or C-section, during feeding. So if the baby is breastfed or if it's uses formula, so different nutrients select for different bacteria. Then you have now the vaginal seeding, which allows for babies who are born by C-section to acquire vaginal bacteria. If the moms have gauzes, if they pass all the STI tests, they can have a gauze in their vagina before C-section, so they can clean the baby when he's out of the womb, and they can acquire a similar community to the babies born vaginally, which is really great. So we know that until 3 years old, the microbiome of the baby is variable. It reaches some stabilization about 3 years old. So until then, it's not very nice to exaggerate on antibiotics and all that, because we want this microbiome to be developing the most closely to the natural one given by the moms. So the establishment of these microbiomes can lead either to health or to disease. Disease status include the bad bugs, Clostridium, Methanobrevibacter, Campylobacter, that lead to leaky guts and inflammation, and therefore different problems like diabetes, IBD, or infections. If the good bacteria are established early on, since the baby is born vaginally and is breastfed, etc., these babies maintain good amounts of bifidobacteria, Faecalibacteria, and Lactobacillus that can maintain the colonocytes very happy, leading to a phenotype of health. So ecological changes in the human microbiome are similar to what happens in the macroecology, right? All species have evolved in the presence of disturbance. And when we have one disturbance, the community can go back to initial states, so can be resilient. But when different stressors, different perturbations occur, sometimes the communities never go back to the initial normal state. They maintain this alteration state. And this also happens in the human microbiome, and that's why we need to identify these disruptions and be able to take them into account when we study the human microbiome. So what we know from the human microbiome is that different niches have different diversity in composition. The more diverse niche is the human colon, followed by the mouth, the esophagus, the stomach, then the skin, and the least diverse is the vagina in terms of the number of species. And when we look at this PCOA here, we see a distinct community between oral and fecal, and then some similarities between skin and vagina. But why are these microbiomes so important? Because they can even protect you from these new diseases. So when we look at this Bach plot with the reduction of infectious diseases through time due to immunization, hygiene, etc., we also see an increase at the same time of autoimmune diseases and multiple sclerosis and diabetes, asthma, obesity. And this is all related. This was published in 2002, where there wasn't really human microbiome studies. But when now, at the light of all this research that has been compounding in the last 10 years, we know that diversity has been reduced in modern societies. With westernization, we see a significant reduction in gut diversity. And this loss of diversity can be associated with the increase in these autoimmune diseases. Today, we know that most of these diseases, like diabetes, asthma, can be cured and are really caused by these microbes. And the disruptors could be, diet, supplements, and medications, and excessive use of detergents, etc. For example, hunter-gatherers have very, very diverse microbial communities in their guts. For example, hunter-gatherers they still heat honeycombs with pieces of wood, so they have spirochetes in their guts. And we know that westernized societies have lost entire phyla like spirochetes. No one of us living in a modern society has spirochetes in their intestines. So we have lost the capacity of utilizing all these complex fibers. And this is, it's terrifying when we look at, the human societies that live more rural lifestyles have higher diversity and are more similar to each other compared to those that live in more urban, modernized societies that have lost lots of groups of bacteria due to these different perturbations, including antibiotics, C-sections, formula, postpartum antibiotics, and even vaginal douching. And I'm adding this here because as I am interested in women's health, you're going to see that many perturbations in the vagina can be really very difficult to come back to a resilient state. So this is one of the disruptors of the vagina. Not only antibiotics, but also copper IUDs, the intravaginal insertion of substances, teas, and herbs, and cleansing with these vaginal douches. The use of soaps and harsh chemicals also destroy the microbiota, with an increase in bacterial diversity in vaginosis. So as I've shown before, the vagina is being the least diverse. We have to maintain this diversity at low levels. So we should not have higher diversity. When we see vaginal higher diversity, we know that the ecosystem is perturbed, just like here in vaginosis. And when we use, for example, these IUDs, you see a reduction in Lactobacillus. So is it really a low diversity organ? It is in principle, because it's the simplest niche. It has an epithelial barrier under the influence of estrogen, so it produces glycogen, and this complex sugar is used by the Lactobacillus to ferment. So the production of lactic acid drops the pH, and by lowering the pH, we have a chemical barrier that avoids the growth of other non-indigenous microbes and anaerobic microbes to live there. Once this chemical barrier is perturbed by either vaginal douches, even clothes that are not cotton, all these changes in the epithelial environment change the abundance of lactobacillus and thus allow for other bed bugs to enter. We also know that different ethnicities, so Asians, whites, blacks, Hispanics, have different amounts of lactobacillus and have different amounts of different groups. So we see, for example, Hispanics have a lot of lactobacillus einers and high diversity groups. So one of the things that I was interested in is understanding what happens to the vaginal and to the cervical microbiome with human papillomavirus infections. And we've seen an increase in this in the last years, especially in young people, in the oral cavity, in the anal cavity, and also in the cervix. 80% of sexually active women are infected at least once in their lifetime by human papillomavirus. These are skin, and they enter the cells by skin-to-skin contact, so it's very important to use prophylaxis. There are about 180 HPV types that have been sequenced, and about 40 of these infect the mucosa and are sexually transmitted. The most important ones and most famous ones are type 16 and 18, which are in the tests. The Papa Nicolau tests indicate that whether you have 16 or 18 or other high risk or low risk types. In our case, we genotype about 25 different types of HPVs. And we know that HPV is one of the causes of cervical cancer. So 99% of the tumors have the HPV DNA in them. And despite the increasing vaccination of HPV in young people, it still remains a significant problem, especially in developing countries and in Latin America. So that's why we've been interested in studying the cervical cancer in Puerto Rico. Puerto Rico has about 20% of the deaths are due to cancer, mostly due to colorectal cancer and other infectious like H. pylori, gastric cancer, but mostly human papillomavirus related cancers such as head and neck, cervical, anal cancer, and also penile cancer, which is very rare, but it's very high in Puerto Rico. And a recent paper, JAMA Oncology, has shown that in Puerto Rico, women from 35 to 64 have increased cervical cancer cases from 2010 to 2017. So it's really a very big problem. So our hypothesis was that high-risk HPV promoting the persistence of this virus could be due to the changes in the microbiome. And in the expression, these carcinogenic pathways could be due to different bugs. So we wanted to look at changes in the cervicovaginal microbiota in the interface between the host and the environment by collecting data from vagina, anus, cervix. We also collect urine and cervical lavages. We have a cohort of about 300 women with IRB and IVC protocols approved. We recruit patients coming to the University of Puerto Rico Hospital. And we also, besides a very long questionnaire on lifestyle, sexual types of sexual behavior, clinical variables, we also do the Papa Nicolau to understand if and to be able to typify the HPV and to see the cytology. And when needed, we also receive biopsy. So the results, if the person has a normal cervix, like here, or CIN1 when you start seeing the first changes in the epithelium, or CIN3, which is precancer. And then ASCUS is when you have small white I don't remember the name in English, but it's not cancer. You can remove this by cryofreezing and it's fine. You're never gonna get cancer. And these lesions here are caused by low-risk HPVs, while cancer and pre-cancer are caused by high-risk HPV. So we collect swabs and we put them in RNA later, and also some of them are dry swabs from the cervix, the anus, and the vagina. We collect urine, and the lavages are basically sterile water, and there is inserted in the vagina to wash the cervical canal, and we collect this water, and then we aliquot this in different solutions to do different approaches. So what we wanted to do is look at bacterial diversity, HPV genotyping, and HPV diversity. Besides 16S, we wanted to look at ITS. We have some shotgun data as well, and we wanted to do cytokine profiling. And this would be also to integrate in a student's thesis. So this is funded by the Puerto Rico Science, Technology, and Research Trust, which is an NGO that funds local studies to be able to compete to R01s in the US. So basically, preliminary data from our studies indicate that women can be colonized by many viral HPV types. Mostly higher diversity comes from samples that have high-grade lesions, which are conducive to cancer. And this can have even 16 different types of HPVs. And we have collaborated with Maria Gloria Dominguez and Daniela Vargas in Venezuela studying Amerindians. And we have found very similar rates of oncogenic HPV types in these Amerindians. What we saw with this HPV diversity is that according to cytology, so high-grade lesions and low-grade lesions, what we see here is that high-grade lesions have higher numbers of HPV. So not only we would expect to see higher diversity of bacteria, for example, but we also see, very interestingly, that people with the biggest lesions, even with pre-cancer, they have the highest numbers of viruses. We had about 52% of people had high-risk HPV, 43% had low risk, and in 21 people, there was no HPV detected. So in terms of the global patterns of bacteria, we find similar patterns across vagina, which is introitus here, posterior fornix, very similar patterns. But then the anus is totally different, as expected, right? Dominated by Bacteroides, Faecalibacterium, Prevotellus. While here in the vagina and the cervix, it's dominated by Lactobacillus iners, other Lactobacillus, Phenagoldius, Snithia, Gardnerella. And then you have low-risk HPV types, which have different, more Lactobacillus and different composition. When we look at the presence of HPV, so either if it's low risk or high risk, we find significant diversity of bacteria in high risk HPV types. And when we look at, for example, in the cervix, even in the vagina, when we look at precancerous lesions, we see here a significant amount of bacterial diversity in CIN3 compared to CIN1 and even to controls. So we show that bacterial diversity is significantly higher, both in vagina and in cervix in women with precancerous lesions. When we look at the ubiquity, this is a ubiquity plot that we developed here in the lab. The x-axis shows the relative abundance of OTUs, and the y-axis is the ubiquity, or the proportion of samples that have these OTUs. And if you look at, I think this is fascinating, when you look at the HPV negative people, you see the Lactobacillus iners has a relative abundance of about 23 to 25%. HPV negatives, no lesions. And when we look at folks with low risk HPV or high risk HPV, you see a drastic reduction in these Lactobacillus iners. So they go from 23% to as low as 3% with HPV. So we know that HPV is associated with a dramatic reduction in the amounts of lactobacillus, especially Lactobacillus iners, which seems to be the protective lactobacillus in the Puerto Rican population. When we look at what are the other important bacteria in the vagina that are associated with precancerous lesions, we find Atopobium vaginae, which is also found in Caucasians, also in precancer and cancer. Gardnerella vaginalis, which is known for bacterial vaginosis. It's very pro-inflammatory taxa. We also find it here. We find Thysiareia preacuta, which is an oral bacteria that is also significant in high-risk HPV. And we found Prevotella bivia. This is interesting. I'm going to show you later some associations of specific bacteria that are from the cervix that are increased in abundance in the oral cavity associated with cancer, oral cancer. And we see here in this case, Thysiareia, and we see, for example, here Parvimonas micra, which is also an oral bacteria that is significantly increased in CIN3 in all the body sites. So when we look at this, each bar represents precancerous lesions. So you have no lesion, CIN1, CIN3, here the same. And you have cervix, vagina, and anus. And we see a significant increase in Gardnerella vaginalis in precancerous lesions in the cervix, in the vagina, and in the anus. Same happening, we have Lactobacillus acidophilus, which for a Caucasian could be a biomarker of health. In Puerto Rico, it's not. Remember, it's Lactobacillus iners. And we see a reduction of this Lactobacillus with CIN3, with precancer. And then Crispatus, which is also a known Lactobacillus that is dominant in health in Caucasians, which is dominant in precancer lesions here. Then you have Prevotella amnii, which is also increasing in CIN3 in precancerous lesions in all body sites. And you have Parvimonas micra, which is not supposed to be in the cervix. It's not a very common keystone of the cervicovaginal environment. It's actually an oral bacteria, which is significantly abundant in the precancerous lesions in all body sites, in the cervix, in the vagina, and in the anus. And this is very surprising. And later on, I'm going to show you some studies where we studied oral microbiomes in head and neck cancer, and we see very similar patterns. When we look at the fungi, we see different body sites. So the introitus, the vagina, the posterior fornix, which is cervix, and the anus. And in the opposite of bacteria, right? Remember that bacteria, we have vagina and cervix very similar to each other, with the anus very, very different. While in this case, fungi are very similar in composition in all three body sites. We have a dominance of Candida, Sporidiobalaceae, and Malassezia. Malassezia is usually more represented in high-risk HPV types in all body sites. So basically, if we were to define the majority of the fungi in the anogenital niche, we find Malassezia, which is more dominant in those with high risk. And we find Candida and Sporidiobalaceae in every patient. So most people have Candida, either if they're healthy or not. But when they're not healthy, they have an increase in Malassezia. Malassezia has been associated with psoriasis, eczema, seborrheic dermatitis, dandruff, and it's also associated with certain tumors. They travel with the tumors. Very recent papers show that Malassezia can be triggering oncogenic processes. And when you look at that in terms of biopsy, so women with no lesions have very low diversity of fungi, but then when you look at CIN1, you see here an increase in those that have high-risk HPV, and in CIN3, you see a significant amount of fungi in these precancerous lesions. So when you go from a healthy epithelium to a precancerous lesions, you increase the diversity of viruses. So you have a lot more viruses. You increase the diversity of bacteria, and you also increase the diversity in these high-risk HPVs and in lesions of fungi. So definitely we go from a simpler niche to a more diverse niche at all inter-kingdom levels, right? Again, we see here the high-risk HPVs have higher diversity of fungi, and when we look at high-grade and low-grade lesions, they have more diversity of fungi compared to women that have no lesions. And when we look at the abundance of Malassezia globosa, one of these lipophilic yeasts that is very associated with skin diseases and triggers these oncogenic pathways, you see the second columns here are high-risk HPV. So the first column is anus, high risk, then anus, low risk. You see a decrease in low risk. Then you have the introitus or vagina, high risk, vagina, low risk, and then the posterior fornix. So in high-risk HPV, in all body sites, you see a significant increase in Malassezia globosa. When we looked at another component of the microbiome, the archaea, and this was done by shotgun metagenomics. This is the short manuscript that we published last year. We see something very interesting. Remember that we were going from biopsy, so no lesion, healthy women, then CIN1 with some epithelial changes, and CIN3 with cervical precancer lesions. What we see here is a significant decrease in metanosarcina in this with the advancement of disease. So healthy women have higher levels of metanosarcina that decrease towards precancer. But then other archaea increase with precancer. So the levels of Methanothermococcus are very low in women without any lesions, but then they increase with CIN3. Metanosarcina are methyllotrophic, so they are known to metabolize methanol and other simple carbons like organic acids. So they concur with Lactobacillus. So they live simultaneously with Lactobacillus, probably living off the lactic acid and that they produce from the metabolism of this Lactobacillus. And then Methanothermococcus, these methanotrophic bacteria, they are heterogenotrophic, which they're capable of producing methane from CO2, and they're strict anaerobes. And we know that strict anaerobiosis and the increase in this kind of taxa is associated with cervical disease. So this is very interesting, and I think it's important to take these studies into account when developing new probiotics for cancer prevention. You cannot only use Lactobacillus because we know that there's other bugs here like Malassezia or these methanogenics that are important. And I think for the future, we need to do a lot of these ecological surveys to understand the types of probiotics that people really need. And of course, you're not going to study a Caucasian or an Hispanic from the US with the same types of probiotics as you would do for a Puerto Rican, living that, their lifestyle and their diet and their sexual behavior is different. So we also have a similar project, which is a cohort of 200 women that self-collected swabs from the vagina and the anus. And we did similar analysis. I'm just going to show you some preliminary data. And again, we see Prevotella being abundant in high risk and both high risk and low risk. Gardnerella, also very abundant in high risk HPV. And then Lactobacillus, very abundant in HPV negatives. Snithia and Atopobium, also very dominant in low risk and high risk HPV. So what this is basically confirms that even when people self-collect their swabs, the community composition is very similar to those collected by the clinicians. We also did some analysis trying to understand if people after menopause, there are changes in the vaginal microbiome. And although we don't see any significant changes, and there are no significant p-values here, we see a little bit higher diversity in post-menopause, a decrease in Lactobacillus, that's clear, and Lactobacillus iners and Lactobacillus gallinarum, an increase in Gardnerella also. But again, this was not very significant. But then when we look at this ubiquity plot of the postmenopausal and regarding to HPV types, we see here the premenopause HPV negative. We confirm again about 25% dominance of Lactobacillus. This is again in the self-collected cohort, showing very similar data to what we showed with the clinician-collected samples. 25% relative abundance in healthy women, no HPV, no lesions, premenopause. But then post-menopause HPV negatives, Lactobacillus reduction is great, as I showed you in the last slide. But then HPV positive, you see premenopause here, you see the reduction of 25 to about 10%. But when you look at post-menopause, HPV positive, that's the greatest reduction in Lactobacillus. You go from 25% to less than 5% relative abundance. Post-menopause HPV positive. So we have to take this into account to better develop strategies and to modulate the vaginal communities of women at different stages. And we should take into account not only the HPV types, but also the age of the women itself. And also we wondered, how about body mass index? There's a lot of obesity in the island. What if women had obese had a different microbiota? Indeed, we eliminated those that had HPV infections. We are only looking at BMI. And when we look at here, obese women have significantly higher diversity compared to normal weight. Even the overweight to normal has very significantly higher diversity. So obesity by itself induces changes in the community composition of the vagina, inducing a higher diversity. And this higher diversity is also accompanied by a lower diversity in Lactobacillus species, especially a significant reduction in Lactobacillus iners. From normal, which is this blue here, bright blue and dark blue, it's Lactobacillus. So you see a reduction with overweight and obesity. And this is also something which may indicate that obesity, not only lifestyle, but also obesity, could be a risk factor for developing vaginal dysbiosis and being more prone to developing to other consequences like cervical cancer. So the proposed model for cervical cancer in this cohort starts by no lesions, having higher dominance of Lactobacilli iners that can be as high as 25%, having Metanocsarcina barkeri, Sporidia wallacei, some Candida. CIN1, you see a decrease in Lactobacillus iners, the appearance of other types of Lactobacillus and of Atapobium and Gardnerellas. And we see an increase in precancerous lesions of Methanothermococcus, Cladosporium, Malassezia. And what we see also is an increase in diversity in bacteria, in fungi, and in HPV genotypes towards disease. So we also wanted to see if we're able, with urine samples, if we were able to collect, to define a metabolome and to try to discriminate the metabolomes from patients who were HPV negative, who had both co-infections, positive and negative, high risk and low risk, sorry, and only high risk infections. And we were able to define a total of 24 metabolites in the urine. And HPV high risk had 3 specific biomarkers: 5-oxoprolinate erythronic acid and N-acetylaspartic acid, which discriminate high-risk HPV from the negative controls. These are metabolites that are involved in a variety of biochemical processes that could eventually be biomarkers for HPV in urine. For example, N-acetylaspartic acid is a family that is involved in the transition of cells to cervical cells. and promoting epithelial tumors. So this could be very potentially interesting in the development of kits to detect HPV infections by the use of urine, like pregnancy tests, for example. So another study that we did, a multi-omic analysis combining cervical microbiota to the metabolites, we showed that there's microbial-derived metabolites, mostly peptide and amino acid metabolism, that suggests an increase in high-risk HPV compared to HPV negatives. And we see here an increase of atopobium vaginae being produced metabolites from L-threonine and acetate, and Lactobacillus iners being mostly associated with the production of acetate, and Shuttleworthia being more dominant in the HPV negatives, producing succinate. And this was published last year. More recently, we have worked on the cervical lavages that we have collected. So remember that we inject sterile water in the cervix and collect these cells. So we developed the same non-targeted metabolomics approach, where we identified 47 with the first 47 samples. We have now about 200 and something samples being analyzed. This preliminary data from 47 samples divided by NILM, which is no intraepithelial lesion, and HSIL, so high-grade lesions. And we see a significant amount of metabolites from amino acids and hydroxy fatty acids from high-grade lesions. And if you see, there's also some columns showing normal weight and overweight. And we see that women that have overweight have specific metabolites that are not produced by women with normal weight, regardless of their cervical lesions. Tryptophan metabolism is increased in women with normal weight and healthy women, compared to low-grade lesions which had higher antranilate metabolism. And also BMI clearly has an effect on metabolite concentrations. When we look at specifically the taxa that is producing these metabolites, we see that, for example, here in high-grade lesions, we divided normal weight and overweight, and you see that overweight women, again, have lowest numbers of Lactobacillus iners and Prevotella, higher amounts of Gardnerella. So per se, a higher BMI is changing the metabolism of the cervical microenvironment, which can lead and facilitate these oncogenic pathways. Another thing that we wanted to look at are biogenic amines. They are linked to malodor of bacterial vaginosis, so the putrefaxine and all these molecules. And this leads to a reduced competitiveness of Lactobacillus. So we wanted to look at what happens if we could detect in the cervical lavages in these women with high-grade lesions, if we could detect any biogenic amine. And what we did is basically detect a higher concentration of these amines in high-grade SIL compared to normal epithelium. And so HPV triggers the consumption of tyramine, which is a genotoxic amine that is linked to cancer development. And the bacteria that is responsible of consuming this are Prevotellas. Prevotellas consume digest tyramine via monoamine oxidase, while Methanobrevibacter, which I showed you before, that increases in CIN3, so precancerous high-grade lesions, they also consume tyramine via tyramine glutamate ligase into gamma glutamyl tyramine. So basically, the degradation of these biogenic amines occur mostly in high-grade lesions by these bad guys that I showed you before in the profiles, so Prevotellas and Methanobrevibacter. To add a bit to this story, we recently were awarded as part of the CAMPO by the NCI, the California, Mexico, Puerto Rico Consortium, to build a consortium to study cervical cancer prevention, reducing HPV in HIV populations in both countries. So one of the studies that I'm in charge in is to see changes in the cervical and anal microbiome with suppositorium that is a probiotic, and we expect to see changes, a reduction in diversity and a reduction, coming back from cervical lesions by the use of this probiotic. So this is a very big clinical trial, and that we are already starting. And another really cool thing about this clinical trial is that we have a machine called Attila that when the patient comes to the clinic, we have a technician in the Attila that typifies HPV in 20 minutes. So the person can go ahead and show the report to the doctor, and the doctor will know that if you have high-risk HPV, you're going to have treatment before you go home. Instead of normally you would do a pap smear, and then you would come to the visit to the clinic 2-3 weeks later. And this way we can treat the patients, collect the samples, and inform them about their HPVs in real time, and also give them if they don't take antibiotics and they pass the exclusion criteria, they can be included in the microbiome study. So it's very exciting. Another HPV, microbiome HPV frontier that we have in our 21 on NIDCR, looking at periodontal disease in high-risk Hispanics. And although we don't have yet the HPV genotyping results, we do have the periodontal assessment, and we have the microbiome. So I can show you some preliminary data from Kim Mill. This is her master's thesis. And she's basically showing that in yellow and red, you see increasing periodontal disease. In green, you have no disease. So you see that mild and severe disease have significantly higher diversity in terms of all men and women together, or only females or males. You see the same patterns, higher diversity in disease. We see an increase in Proteobacterium and Verruco microbia in mild disease. And the biomarker for severe periodontitis is Prevotella. This is a cross-sectional study with 134 participants so far that have been analyzed by the microbiota. Apologies. And some of these taxa that she shows to be dominant in severe periodontitis include Prevotella, Fusobacterium, which is also been seen as a very bad bug in colorectal cancer, for example. Treponema denticula is very dominant also in severe periodontitis. And then you have Atopobium. Atopobium, as I showed you before, is a cervical bacteria, and you have it here in severe periodontitis. This could be linked to oral sex, because this bacteria is not supposed to be in the oral cavity. Then you have Parvimonas micra, which is also very dominant in severe periodontitis. So, and with this HPV periodontitis study, one of the things that we have also, it's a very large questionnaire with metadata, so that we can understand different types of behavior in relation to their microbiomes. But the things that people mostly don't answer, and we have tablets and people use headphones, we don't interfere with our questions and answers. But even though their responses in relation to the sexual behavior are very rare. For example, we ask how many times they have given oral sex to men, received oral sex from men or women, and they prefer not to respond. These are very sensitive questions that sometimes people lie, even though we are not close by or we don't interfere in their answers. But still is something that made me start another study with the rhesus macaques, specifically because of the promiscuous behavior. So one of the things that that is tied with this taxa that we've seen in periodontitis and taxa that we've seen in increased lesions in the cervix, we have also seen this in head and neck cancers. So we have published 2 papers with collaborators from let's forget. It's a university in the US. Never mind, I'll remind me. I'll remember later. Johns Hopkins. So this was it's a Johns Hopkins cohort of patients with head and neck, and they were Caucasians. 34 had cancer, and then we compared with human microbiome project, saliva samples, mid-vagina and vagina, and we found so interesting that folks with head and neck cancer had a bunch of different lactobacillus, and in the oral cavity you don't have this many lactobacillus. Mostly you don't have Lactobacillus vaginalis or Lactobacillus johnsii. And where do we find it in the human microbiome project? We find it in the mid-vagina, and we find it in the introitus. So we see that Lactobacillus is 700 times higher in head and neck compared to the healthy controls that basically have no Lactobacillus. And this is an indication that oral sex is bringing Lactobacillus from the vagina to the oral cavity, and they're changing the epithelial environment, and they're triggering oncogenic processes in the oral cavity. And it's also other bacteria do that, like Fusobacterium, which is also very dominant in periodontitis, as I showed you. Lactobacillus, as I mentioned, Papillomavirus micro, which is also very inflammatory, but it's an oral bacteria. Another similar study, we did a study longitudinally looking at the same patients over time, and those that had head and neck cancer had Prevotella and had in green the Lactobacillus. Again, another study showing that Lactobacillus bacteria is dominant even longitudinally in head and neck cancer. And in terms of the Papillomavirus micro, and this is the tie to the cervical cancer study, we see Papillomavirus micro in head and neck compared to controls. It's significant abundance in the oral cavity, Papillomavirus micro. But then you see also Papillomavirus micro being significantly abundant in the cervix, in the vagina, and in the anus with precancerous lesions. And this is, as I said, Papillomavirus micro is an oral bacteria. An oral bacteria in the cervix, vagina, and anus in high dominance in precancerous lesions is also telling us the opposite, right? So this is bacteria from saliva. So oral sex can be determinant to oral and cervical cancer. And since this is very difficult to study in people because people don't feel comfortable answering specific questions about sexual behavior, Luis has started his master's study and doing analysis of papillomaviruses in a population of rhesus macaques in Cayo Santiago. As I mentioned before, it's an island where free-roaming rhesus macaques have been there for more than 50 years. So you can go ahead and collect samples from there. So he started collecting from the oral cavity, from the penis, from the penile, inside the penis, in the surface of the skin of the penis, and also in the anus and in the vagina. So he has all these samples, and he wants to look at microbiome dynamics and source tracking, since these animals have promiscuous behavior, and this would allow us to better understand if the route of oral to anal, as we expect occurs in humans, occurs also in these animals. So he wants to and he also does this with conventional and SPF, so animals that have been pathogen-free. He does this in these two groups of animals. And now we have a recent study on SARS-CoV-2. We have a big cohort of participants, including healthcare workers and pregnant women coming to the hospitals. We've been collecting blood, saliva, oral swabs, and anal swabs to look at association between oral and fecal microbiota with clinical manifestation of COVID-19. And also our aim was to help, to bring more PPE to the hospital and to educate the community, and also have do tests. So we do the SARS-CoV-2 tests in saliva and in also nasal swabs. And we wanted to have this available to the community, and the biorepository is also available for whoever wants to do these studies or to look at other molecular markers. So it's open. If any one of you is interested in collaborating, just write me. So for today's talk, I focused mainly on the cervical microbiome. You know that there are not good or bad guys. They're everywhere, but they're focused in the niches that they are more suitable and they're more helpful in. So the vagina has specific bugs that protects it from the invasion of other serious pro-inflammatory bugs. So we want to have it simple, low pH, dominated by Lactobacillus. Although we know that Hispanics have a bit less Lactobacillus than other ethnicities. We know that HPV infections are related to changes in the epithelial microenvironment and to changes not only in the community composition, but also in the metabolism, and that cervical carcinogenesis is favored by an increase in this on the diversity of both bacteria, fungi, and also HPV genotypes. We have identified Malassezia being a very bad yeast that is associated with precancer, as well as Methanothermococcus, compared to methanosarcine in the healthy people. We have seen that HPV infections triggers also the metabolism of biogenic amines, which give the bad smell to the vagina. And this is associated also with species that are very anaerobic, like Prevotella and Methanobrevibacter. We have seen also a link between oral sex in the development of head and neck cancer, periodontitis, and also cervical cancer. And ultimately, what I wish is to understand the rules for this community structure interkingdom, so with bacteria, with archaea, with fungi, and to be able to manipulate these microbiome components towards health in order to develop new probiotics and new tools that could stabilize this microenvironment to favor and to prevent health. And my last slides are to, as a member of the Microbiota Vault, to call attention for this tremendous humanitarian effort by Dr. Dominguez Bello and her team in order to conserve microbes from biodiversity peoples. It's very important that we teach even how to study microbiomes in the developing world, because we need also to make sure that they trust the storage of their samples and they are involved in this approach. Microbiome has become universal, and now with the COVID pandemic, everyone is giving their talks and sharing their science. That we need to be able to share tools, to share scripts, to share data, to share collections, and we are going towards an open microbiome field. And I'm very proud of this effort. And also, I wanted to bring your attention to the ISME Latin America, the second meeting. First one was in Chile. It was amazing in 2019. For those who were able to go, I'm sure it was memorable. We wanted to do the same now in Bogota, but it's not going to be in person. It's going to be online from 27th to 30th of July this year. You can go to ISME Latin America and Facebook. Here's the link. Also, you can send me an email. And in Puerto Rico, we're organizing the Microbiomes Symposium from Environment to Human Diseases. We'll have Greg Caporazzo, Anand Lande, Keith Crandall, and Kerry Keeney from UTexas, and they're going to give different aspects of their own microbiome research. So it's going to be very exciting, and you can register in our webpage. You can go to our webpage and register, or again, if you want more information, you can email me directly. I just wanted to thank my funding agencies, the Puerto Rico Science Trust, the HIREC, the Alliance for the Advancement of Biomedical Research, Puerto Rico IMBRE, MyR21, and my great collaborators, Dr. Josefina Romaguera, Dr. Larry Forney, which is a mentor, Natalia Chorna, the biochemist specialist, Ana Patricia Ortiz, Carlos Ariol, and all my great students. And if you have any questions, feel free to email me. I'll be happy to communicate with you. Thank you.
Thank you very much for your nice talk. That was really fascinating. So we do have a few questions.
Are we good on time?
Yes, don't worry about time. It's all virtual. Time is just a concept. So one question is, have you looked into whether there is increased risk in cervical cancer or any significant changes in microbiome relative abundances in individuals that are undergoing hormone replacement therapy? You know, like either post-menopausal or gender affirming.
Yeah, we have that metadata. I have not analyzed the data, but I will certainly analyze the metadata from the self-collected cohort, and we're going to include it in this. Certainly, I cannot say anything at this time, but we will include it here. Yeah, the menopause. Yeah. So this we see an increase in diversity. It's not significant, but if we divide this by hormone replacement therapies, maybe we could see something, but I'm not sure. I haven't done that analysis, but we will certainly do that. Mm-hmm. Cool. It was also requested by the reviewers. So.
Maybe it was the same reviewer.
We never know.
I was also wondering, so when you, in the beginning of your talk, when you presented about like how life in humans starts with when you're born and then you have like the vaginal microbiome, I wonder, do you have any evidence whether there is any kind of microbiome that is transferred from the father? You know, like microbes in the sperm, or also at least some like heritable paternal effects on the offspring microbiome?
That's actually a good question. I wouldn't think so, because I believe in the theory that we are sterile in the womb. We are born sterile. If anyone has found bacteria in the placenta, and there are a couple of recent studies who show 16S profiles in the placenta, I think that's, it's DNA that is there from macrophages that have, you can amplify them by PCR, but that doesn't mean they're live bacteria there. But I think that during birth, that's the first contact that humans have with their microbiomes. Before, we are sterile. So during development, of course, we are interacting with the parents that are at home, the animals, the environment. So we are co-colonized by all these bugs. But in the womb, I don't think we have an impact of the sperm in the zygote. No, I wouldn't think so. No.
Interesting. Then I have a follow-up question to this, which is, what is the relative importance of the vaginal seeding microbiome for the baby? Because I would assume that breastfeeding and cuddling with the baby during the first couple of days might be more important, as babies are often also cleaned after birth. And
if your baby is born by a vaginal delivery, you don't have to worry. You just have to make sure that they are in close contact with the other colonies of lactobacillus that you have in your body. The breasts, they have some certain creases when the milk goes out, right? And the breast pezon, I don't remember the name. So there are populations of lactobacillus that are different from the vagina that live in your pezon. So even if you don't have milk, even you formula feeding your baby, you should always try to latch your baby to your breast so that it gets colonized by this specific lactobacillus that live in your breast. That's one way to do it. Another way is to supplement. If your baby was born by a C-section and you haven't colonized your baby with the gauze because that trial is not available in your hometown, you should supplement your babies with probiotics. Definitely, there are probiotics for kids, and you should when they enter solid foods, you should give them fruits, and early on you should give them fibers so that the lactobacillus they have in their guts, they can thrive on these prebiotics, on these complex fibers, and this should continue during life. You can come back from obesity, from IBD, from a lot of problems by eating a lot more fiber.
Mhm.
Because that no fiber and probiotics, even if you have a probiotic which is like 30 billion colonies, if you don't have any fibers, if you eat pizza all the time, if you are stressed all the time, those probiotics they go with your fecal matter, right? They're not established. But if you eat fiber, you have more chances that they will thrive in your gut.
So that means you're actually also kind of comforting women who maybe had a emergency C-section or something, that as long
I did, I did, and it's fine, and it's okay to give formula, but it's always and all these studies came after my baby is 14. She's not a baby anymore, but I breastfed her for 4 months, and I didn't have a pesson. I apologize, I don't remember the name in English, but I had to use a plastic device attached to my breast so that she could suck my milk, right?
Mhm.
But still she had she had even later I took it off, and she was attached to it. And one thing, I'm Portuguese, so we give a lot of soup. Even though she was born by a C-section, she has been thriving on fiber. I eat soup all the time, cabbages and uh, onions, carrots. We eat that every day, so that's very important. And even though she was born by a C-section, she has no asthma, she has no autoimmune disease or anything like that. So it's fine. That's hope at the end of the road.
So then let's switch to another topic. So do you have similar studies on microbiomes of Afro Latinos, or are they included in the presented studies as well?
No, they're not included. They're not included here. We wanted specific to take into account the people coming from the from our hospitals that are mostly Hispanics.
Okay.
We have other regions in the island where Afro-Caribbean people come, and there's another interesting group to include, but in this study we don't have those. Yeah.
Okay. Have you seen any correlations between the use of antibiotics and HPV infections?
So in this specific study, we are excluding people who take antibiotics in the last 2 months
Mhm.
because they can impair the microbiome directly. So we wouldn't see an effect of HPV.
Mhm.
But in the new clinical trial, Campo, we will include everyone with and without antibiotics. So I think in 1 year and a half, I can give another talk and talk about Campo, and I will be able to answer that question. But for this study, we have excluded everyone who takes antibiotics.
Okay.
That's why recruitment is very slow. This project has been around for nearly 4 years, and Hurricane Maria came. We lost all the collection. We had to start over again in 2018, but still the recruitment is not that quick because people here take a lot of antibiotics, and we have to exclude them. So.
Mhm. The next question is, can PIV intercourse with or without condoms lead to changes in vaginal pH and then microbial abundances as well? Maybe that contact with the partner could lead to changes in the seeding microbiome.
Yeah, that's interesting. So, in regards to the first part of the question was the
Well,
pH, the pH, right? So,
yes,
in terms
yes.
of the pH doesn't change much. In fact, Hispanics have high pH. For example, a Caucasian, they can go up as 6. We have like 6, 6.5, even 7.
Mhm.
It's super high for example, in a Caucasian, which is like 4.5, 5, the most. And Hispanics is very high. And that's normal. You can be healthy without HPV, without lesions, and you can have a high pH, because the lactobacillus are also different. But we don't see with the use of condoms, you don't see changes in pH. You do see changes in the microbiota, but still it's the best way of protecting from human papillomavirus infections, because those are from skin to skin contact. So that's the best protection for against this STI. And of course, getting the vaccine. If you have a teenager, vaccine your teenager. If you're an adult, you can also. I vaccinated when I started studying HPV. I was not vaccinated in my time, but when I started, I vaccinated myself and I paid for the vaccine. I mean, because you never know if you have been exposed to these high numbers of high risk types.
Mm-hmm.
So it's always good to vaccinate, even if it's late.
Mm-hmm. Did I understand it right that hormones actually make the vaginal environment more acidic?
It depends. It depends on the hormones. It definitely changes, and women after I think there's certain hormone therapies like plant hormones that are very fashionable nowadays. They help your skin and whatever, and those are really bad because they are really acidic, and but they lead to cervical bleeding. I've seen a lot of cases, and friends of mine that have taken these things for a while, and they've had a lot of problems. Yeah, I think the best thing is not to take anything, just to do exercise, eat well, drink plenty, and accept your time. Be happy with who you are.
So in your studies, the ladies who were self-collecting swabs, did you control whether they took birth control or not? So whether they were taking the pill?
Yeah, in this case, they were not having birth control.
Okay.
Yeah.
And also another question was, how does obesity affect the vaginal environment and ecology? Because you mentioned there were these significant effects of the BMI.
Yeah. We don't know the mechanistic effects, and that's something that we want to understand better. But definitely, when you look at a person who has no HPV, who has no lesions, just the fact that it's obese compared to normal weight, you see a significant reduction in lactobacillus. So just being obese already puts you at risk of developing vaginosis or other dysbiotics symptoms. Yeah, it's terrible.
Yeah.
It's like an antibiotic, or like it's like it's leading you to precancer.
Mm-hmm.
Obesity in the vaginal microbiome.
Interesting. Okay, so thank you very much again for your talk and for your time to answer all the questions.
No, thank you for the opportunity, and I hope that in the near future I can come back with more data, specifically from the RISA study, or even you can invite my student to present. It would be good for him. All right, thank you so much. Thanks, everyone.
So I